simultaneous translation, but we will have the Indonesian language captions for you ready. Okay, good afternoon, everyone. Thank you so much for joining. I'm really excited to see so many people here. Today's webinar is organized by Luminocean together with Ocean Gardener. And in today's presentation, Rahul will speak about daytime coral spawning, which fits very well into our webinar series within the COSMARINDO network. So the Coral Spawning Monitoring and Awareness for Restoration Network Indonesia. With COSMARINDO, we bring together local researchers and coral reef practitioners in Indonesia to enhance the understanding of coral spawning patterns and to promote non-invasive reef restoration methods for those who don't know yet. And COSMARINDO is funded by the Global Coral Research and Development Accelerator Platform, CORDAP, and is initiated by Ruhr University Bochum, Luminocean, Unseen, and University of the Ryukyus. And this is all run in collaboration with University Banda Neira, Universitas Patimura, Universitas Samratulangi, Universitas Hasanuddin, Universitas Udayana, and Universitas Mataram, all in Indonesia. And we are now in the second year of the program. Last year, we conducted preparatory online workshops, on-site coral identification workshops, and coral spawning and larval rearing workshops at the Banda Islands. And beginning of this month was the first time where all university teams that had joined the workshops monitored by themselves at their sites. In addition to that, many more of our network partners conducted monitoring, and I see many of you here. So I thought I'd take the opportunity today to just give you two figures of information of what was going on regarding coral spawning monitoring in Indonesia this month. So the first is the extraordinary effort that all of you contributed. There were in total 17 sites across Indonesia monitored, from Banda Aceh in the west to Raja Ampat in the east. And we had 109 team members in total monitoring. There were 63 night dives conducted across sites and 89 mornings with coral egg trap checks. And what I think which is really extraordinary, 378 hours of training students. And we also saw coral spawning. There were in total 51 nights with coral spawning across sites. So one night at one site would count at one site night. And 79 species that spawns. Whereas here on some sites there might have been an overlap. So again, it's like site species that spawned and 265 colonies that spawned and even 11 spawn slicks that were seen at night. And this resulted into 218 data entries into our joint platform CoralLog, where we collect coral spawning observations for everyone across Indonesia. So I just want to thank everyone who contributed to this. I think it's really huge. And here you see everyone who is part of the network who contributed this month or contributed previous months. Thank you very much, everyone. And what we have also done through this program is we just recently published a framework to teach coral spawning monitoring and coral identification. And this is available now for everyone to download in Indonesian and English. And I would kindly ask Priska to quickly show you or explain to you where you can access this framework that was mainly Priska's really, really huge and amazing effort.
Yeah, thank you, Marika. So I'm going to share with you this framework that we put together, actually. So this is so we will share the link with you. Maybe Malika, you can copy the link to the chat box. And if you click on the link, it will bring you to this page. And this framework is available. You can download it for free. It's available in Bahasa Indonesia and also in English. And so I will just show you a little bit. This framework, we designed it for the universities in Indonesia because we collaborate with universities in Indonesia. So for them to teach coral identification and coral spawning monitoring. And in this framework, you will see that we designed it for 1 semester. So 1 semester is 6 months in Indonesia. But if you are not a university, if you are an organization and you want to teach coral identification or coral spawning monitoring, or you want to just share with your staff, you can also do the schedule that we created here, that we actually do for our training in Banda. So it's like a condensed 3-week training of combined coral identification and coral spawning monitoring. And so there is also a chapter about useful links. So basically, we put all the resources that we use in for our trainings and workshops here, and you can download like all the slides and so on, and you can just adjust it according to your needs. And there is one resource here, the field guide for the corals in Banda, that is written by our coral identification expert Douglas Fenner and co-authored by our coral identification specialist in Luminocean Fabrian, but it's not available yet. It's still going through the finalization process, but once we publish it, the link here will be activated. So don't worry, this for now it's not working, but later it will be working. And yeah, that's it. So you can download it. Go ahead, click on the link, and then you can just use it or change it. Or if you have any questions, you can just send us an email or just use the Coralab platform. There is a contact form. And if the resources also are not working in the framework, we also put it here, and you can download it from here. Thank you.
Thank you, Priska. Yeah, then I would like to hand over to Vincent now from Ocean Gardener to introduce our speaker for today.
Yes, hello guys. Hope everything is all right with you. You're feeling comfortable. We're gonna have to enjoy a very good presentation from Dr. Rahul Mehrotra. He's a marine biologist and ecologist, originally from the UK. He has worked on marine biodiversity and conservation initiatives across the Indo-Pacific. Over the past decade, he has been based primarily in the Gulf of Thailand, having since co-created the Independent Marine Conservation Research Center, known as ATMEC. His current primary area of research is focused on how to best inform coral conservation projects by using the powerful potential of coral reproduction to support reefs today and paving a path for having reefs tomorrow. His other key areas of investigation include bridging the gap between the taxonomy and ecology of various marine organisms, from sea slugs to cryptic corals, and utilizing citizen science and community-driven projects for marine conservation and biodiversity management. Rahul has long been a keen advocate for science communication, especially working to develop a deeper understanding and appreciation for marine life among the recreational scuba diving communities. Most recently, Dr. Mehrotra has joined the team at the Mero Foundation in Bali to advise on conservation and ecology-focused efforts using their state-of-the-art marine laboratories. So here it is, the legend, Dr. Rahul Mehrotra. Please go for it.
Nobody's called me a legend before. Thank you very much. Just give me a quick second. I will share screen while I recover from that introduction. Let me have a look. I will require at least one person to vocalize enthusiastically once the screen is actually being shared. Sounds good. Sounds good. Okay. I'm gonna take a quick second to catch a breath and move all of these Zoom things away from my slides. Okay, so thank you guys for attending, and thank you very much, Luminocean and Ocean Gardener, for organizing this and inviting me to talk about these things. When I say these things, I am going to talk about the daytime spawning, which is, spoiler alert, what is on the screen in front of you. But because I am aware that we have a very wide spectrum of attendees in this call, from people that have never seen coral spawning to people that see a lot more coral spawning than they anticipated, I will probably start with a fairly general and loose intro to things. Please be patient if you're familiar with these things before we get into somewhat more technical and then philosophical aspects. So with keeping time in mind, I'll jump straight in. We're going to talk, as I said, about the basics of coral reproduction, some of the terminology that I may or may not remember to use later on in this chat, talking about predicting coral spawning and the ways that that has changed and what we've been doing to develop a deeper understanding of coral spawning timing here in Thailand, where I currently am. Some quick notes for people, just because people ask all the time that are first stepping into the world of coral spawning, what are the things to look for if you are a recreational diver and things like that. Again, this is a very, very biased and simplified perspective, but I'll put it out there for those folks. And then I'll focus on the Thailand story, specifically the Gulf of Thailand, where I had done more of my work. And for those that are still awake at the end, I'm sure there might be some questions, but you know, we'll see how we go. So with that in mind, let's talk terminology very briefly. Let's see how AI translates some of these terms. So visually speaking, when we talk about coral reproduction, we can talk about both asexual and sexual coral reproduction, and we are going to be talking about the sexual reproduction cycle in this sense. Asexual reproduction is what a lot of people, particularly in the world of coral restoration, are more regularly familiar with, where you get a coral that can, you know, in multiple fragments grow up into mature colonies, or how a single colony grows over time, new polyps emerging from the coral itself, through the process of effectively cloning, how a colony, despite being made up of multiple animals, remains genetically identical to one another. Sexual reproduction is the coral investing large amounts of metabolic energy to produce sperm and eggs in one form or another, that are then released to create new genetic individuals through the combination of gametes, sperm and eggs, from multiple colonies. And this largely happens externally, not always. We'll talk about that in a second. And then once the gametes are released, the fertilization happens, and then fast forward a series of events, you get baby corals. If you want to deep dive into that fast version, then we can talk about that later. So taking things a little bit nerdier, these are a couple of terms that people come across fairly often when we talk about coral spawning, which is the idea of gonochoric versus hermaphroditic colonies. The word gonochoric refers to colonies that are predominantly producing sperm or eggs, you know, often simplified to either male or female. And hermaphroditic colonies are colonies that produce both sperm and eggs. And of course, this does not mean that they can self-fertilize. Put an asterisk onto that if you'd like. And so there is, this is an important thing for us to understand in terms of how the fertilization can happen and various other aspects associated with that. A couple of other terms, brooding versus broadcasting. Broadcasting corals are the corals that will create the gametes and release them externally. These tend to be synchronous gamete release, so coral spawning events. When we talk about a coral spawning event, that is largely talking about a synchronous release of gametes by multiple colonies of the same species, or multiple species within a genus, and multiple genera across the reef. The gametes, the sperm and eggs, are released. They positively buoyant drift to the surface, taking a three-dimensional space into a two-dimensional space, maximizing the chance that any single sperm can meet the right egg, or vice versa, and, you know, fast forward baby corals. Brooding is the analogy would be a coral getting pregnant. Again, I am oversimplifying here, but this is where the development happens internally, where male colonies will release sperm. And if it is, for example, a gonochoric colony, and the sperm will drift across the reef, and then the female colony, in this sense, the egg-producing colony, will capture the sperm, and the fertilization will happen internally. Early development will happen internally, and then a more mature and more developed planulae or larvae will be released by the coral, and therefore it will not go nearly as far. It tends to be much closer in terms of dispersal, whereas broadcasting corals, you can get dispersal on dozens, if not hundreds of kilometers, whereas in brooding corals, that tend to happen on the scale of dozens or hundreds of meters. And so there's a whole range of aspects associated with that we can talk about later, maybe. These are some simplified versions of these terms. Let me take away from the text so people can just look at some photos of spawning. This is mostly just to give people a break from the text. On the left, we have one of my favorite spawning corals to look at underwater, Galaxia, top left. We have Cephastria on the bottom left, and then we have Goniastria on the right. And so, yeah, for those that have, for some reason, not seen coral spawning anywhere online, we need to change your social media algorithms to see more of these things because they're very cool. So I just want to quickly touch on a theme that we'll talk about, which is challenging the preconceived understanding of a lot of coral spawning, which emerged, a few decades ago, particularly with work done in the Great Barrier Reef, where we had an understanding of coral spawning being, strictly either brooding or broadcasting or gonochoric or hermaphroditic, and versions of that would apply, across the known range of corals. But I'm going to talk a little bit about Diploastria from work that was done by James Guest and others over a decade ago. And instead of explaining this, I'm going to just read what they found. Oops, I'm going to just read what they found. In this paper, they showed that in Singapore, Diploastria is a broadcast spawner. So for those that are not familiar, these are terms we know, with predominantly gonochoric polyps. So the individual polyps themselves are either going to be producing sperm or eggs. Colonies, however, contained both male, female, and a low proportion of co-sexual polyps during their sampling. The most plausible explanation for this is that polyps switch sexes with egg development and sperm development cycles occasionally overlapping. And this leads to colony level alternation between breeding seasons. The reason I'm quoting this verbatim is this is one of those studies that I love to mention because it starts to break the rules that we had been using to understand. I say we, this paper predated my work with corresponding, but within the community of trying to put these things into boxes, those boxes and those barriers started to get a lot more blurry. And once we started to add either more locations, more coral taxa, and the rules within which we understand coral spawning, the times of the year, for example, all of that did change and develop. So talking about predicting coral spawning events, touching closer to what is the actual theme of the talk today. Conventionally, a lot of coral reproduction and sexual coral reproduction has been investigated using, again, simplifying essentially histological practices where you would effectively break a part of the coral colony at the right part of the coral colony to look at the mesenteries and to look at basically the internal structure of the polyp to investigate the development of the eggs, in particular, of the gamete bundles. And is a technique that is destructive, but in the past, this was not only one of the easiest and most informative ways to understand the development cycle of a given species within a given location, especially because it's very visual. So very quickly, let me show you some photos of that. Other than the terrible white balancing, these are not my photos, but they are also my favorite photos to demonstrate this. They involve, as I said, breaking a coral, and in modern times, there are many reefs and many species within those reefs that we can no longer afford to do widespread examinations of this because if you're trying to understand the reproductive development, for example, of Acropora on your reef, but now in 2026, there are only 10 Acroporas on your reef, you are now affecting a significant percentage of the total population at your site. And so there are considerations we need to make in terms of how we use some of these pigments. But in this photo, just to show you, the photo on the right, we see, I don't know if people can see my cursor, but we see the outer part of the skeleton where the actual correlates themselves are. We see the pink egg bundles. I like to describe it as chewed up bubblegum. And then we actually have the skeleton underneath. And so the pink fluffy stuff here, or in this case, on the acropora on the left, the round pink gamete bundles tell us that these corals are ready to spawn basically imminently. If they were pale yellow or white, for example, that would suggest that there's still potentially some development to be done. Again, I'm taking a lot of complexity and I'm trying to distill it down. So this is how things had been done in the past. If you wanted to investigate roughly when your corals were spawning, you could go and sample a bunch of these colonies. And this is still a practice, by the way, that is done the world over. But that is a way for you to figure out, okay, the moment I start to see gamete bundles and I can understand the degree of their development, I can understand when the coral spawning for this species might be at this location. There are non-invasive methods to try to understand the timing and capacity of your coral spawning. To the ones that I'll talk about briefly, and the ones that I love to think about are settlement tiles and just in situ monitoring. So for the reasons that I just mentioned, where we can't always afford to investigate internally at the cost of the colony's sort of temporary health, metabolic health, we can use other practices to try to understand output. So settlement tiles are essentially you're studying recruitment rather than spawning itself. But if you put down a bunch of settlement tiles, which are often just small bits of ceramic tiles, sometimes made of other materials, onto a reef, and you can then investigate, perhaps every couple of weeks, every month, for a given period, the settlement of juvenile corals, you can then work your way back to figure out, okay, well, which periods did we not see any corals and which periods did we find some corals? And it gives you a very rough window when there might be some reproductive activity. This depends on how often you look at the tiles. It depends on to what degree you're looking at things, but it gives you an idea. You're tracing back the spawning time based on the juveniles that make themselves visible. Or one can do in situ monitoring. And this is very much the technique that is the most accessible, I think, to a lot of people, which is just to go diving and look for something that will give you that piece of information about the coral spawning time. Now, one does not just do this blindly. You should be working with data to tell you, roughly speaking, this is a probability that I might see some spawning. In an ideal world, of course, we would just have eyes 24 hours a day, 365 days a year, and we would learn so much. That would be phenomenal. But that is, as far as I know, nobody's currently willing to do that. If you are, please let me know. Send me your CV, and that would be fantastic. So here's some photos very quickly of the settlement tiles currently in use from some work done a few years ago, not by our team, and the dispersal aspect of things we'll talk about a bit later, but essentially, as I said, once you have had your coral spawning event happen, you have your fertilized larvae, they swim across a certain distance. I say swim, they are carried by ocean currents across a certain distance before they will either settle or they will not, and they will end up dying in the water column. But recruitment of juveniles therefore allows you to understand which taxa, to what degree, and frequency, so on and so forth. The amount of work that's been done on recruitment in Thailand, where of course I do a lot of my work, is limited, or has been limited for the majority of the coral reef biology period, like academic period of Thailand for the past 40 years. But in recent years, that has dramatically started to change. Okay, let's start to get to a little bit more in-water spawning stuff. For those that are confused perhaps if they have seen coral spawning before, or you're on the night dive and you're not sure what's going on. There are far better tools, by the way, than the slides and the brief version that I'm going to explain in this talk, but just to give you some visuals. A lot of corals, not all corals, a lot of corals, before they actually release their gamete bundles, undergo setting prior to spawning, which is basically the individual polyps taking the developed and prepared gamete bundles and pushing them closer and closer to the mouth prior to release. And that becomes visible to divers underwater if you know what you're looking for. And so that's often visible as white, yellow, or more often pale pink bundles just sitting under the mouth, slowly becoming more and more visible as the coral pushes it closer to its external surface. So on the left, we see Platyjara, right, we see Goniasteria. Here are some more photos of different taxa. We have some varieties here, more Platyjara. On the left, we can see Acropora spawning, and obviously you can see some individual coralites that still have the gamete bundles just ready to be released, but not quite. So setting would look basically similar to that. It varies across multiple genera, but I'm just showing some photos as examples here. So in each one of these photos, if I was to see this underwater during my spawning dive, I would assume that within the next, say, half an hour, other than Platyjara and often varieties that just like to keep you waiting, they will be ready to spawn fairly soon. We see with things like Lobophilia, it's not always the most apparent, not only because the gametes, the eggs and sperm, the egg bundles are very, very small, but you can often see a cloud of the sperm being released by the Lobophilia upon spawning, but it doesn't always do that. And the photo on the right is just an ego boost for me because I love this photo of Galaxia just before it's ready to go. Galaxia is really quite a stunning coral. And you can just begin to see those egg bundles being pushed towards the mouth. So one of the questions we get quite often is, oh, I think I saw some spawning, or I think I saw the egg bundle ready to go. I can see these round things on the coral. Keep in mind, corals are animals. They do eat and they do have mouths. And please try not to get confused by the round things that you see on your corals. Sometimes that's just a coral being a coral, and that's totally fine. There is a difference between seeing the mouths of the individual coralites versus seeing the gamete bundles being ready to be released, what we call setting, as I said. They do look different. You'll see a pigment difference. Yeah, and that should help. For the kinds of things that we are currently interested in at a baseline level, of course, understanding the timing. Again, I'm thinking about this from a recreational dive community perspective that want to get involved in things like that. For the coral specialists in the audience right now, please disregard this. It entirely depends on what you want to know. But the time of setting, the timing of spawning, which species, which genus, or at least tell us it's a coral. Right now, not particularly focused on crab spawning during coral spawning, but of course it could happen. A timestamp, getting synchrony between your dive computer and your camera to make sure that the time is actually correct for those that travel a lot. If you've just visited Japan and then you get into a coral spawning dive, please make sure that your time is actually set to the correct country, unless of course you are looking at coral spawning in Japan. If possible, a count of the number of colonies that are reproductively active versus not helps us understand the reef-wide reproductive activity. And something that is very important, and at least for us here in Thailand, I imagine it's better enforced in other places, I hope. But it is very important to figure out the impact of illumination. When we are down there doing a night dive with our white lights, we are actively introducing an artificial variable at a very sensitive time for corals. This is not something that has had a huge amount of study, but for safety purposes, do try to use a red light where possible. Again, it is a wavelength that dissipates more quickly or doesn't penetrate further in the water column and is likely to be far less of a stressor to corals. But there's a lot more work to be done on this. In aspects of the Gulf of Thailand, this is not possible for reasons that I will, again, spoilers, explain in a bit. A couple of other things. coral setting can happen over quite a period of time. Coral spawning tends to happen over a few minutes to, sometimes 10, 15 minutes, depending on how enthusiastic the coral is. If you think you've seen some spawning and the something is following your torch, that is not coral spawning, that is just something else in the water column, etc., etc. Okay, let's talk about the actual story that we're here for. And I'm going to start by going deep into the context world to explain what the daytime coral spawning and what our work actually built on. So I would say the most well-established start of trying to document coral spawning in Thailand, or coral reproductive activity in Thailand, came from this work by Ajahn Nong in Thailand, where her and her team investigated at the time what was Favia, so Dipsastria, looking at using the same technique that I said, investigating the development of gamete bundles, so breaking open a coral, looking at are they mature or immature, or in simple terms, are they pink or are they white, or in between, across hundreds of colonies over a couple of years in 2007, 2008, across multiple parts of the country. So these are different provinces in Thailand. And we can see that thanks to this work, it was possible to pin down that, roughly speaking, February, March, and April, depending on which coastline of the country you are in, is going to be your peak coral spawning season within the year. So now we start to understand, okay, in Thailand, if we're looking for coral spawning, this is going to be the time of the year that corals are most reproductively active. It lines up well with other Northern Hemisphere spawning observations. Again, there's a bunch of nuance, which is what a lot of this talk is about, but as a general. So taking that a little bit further, a few other studies started to look at coral spawning either in a coral species-specific manner or a reef-wide observation manner. Our work started on one location in the country back in 2000 and something, 14, maybe, 13. But this is a snapshot on the right from work that we did post-COVID, because we had to do something. And I want to explain what we did, which to really start to talk about the things that we don't know. So having identified the coral spawning season and other people's work identifying, okay, it's this number of nights after the full moon that you're more likely to see the coral spawning, and these are the taxa that can be reproductively active at certain times. You identify a baseline for what is the reproductive period for the country. However, that often tends to bias things a little bit, because you are extrapolating conclusions based on a single investigation on a single, in this case, genus or a couple of species, and extrapolating that to mean that all the corals on the reef. But that is not necessarily the case. In a lot of, this is what I was referring to when we talked about the Diploastria work, where as you widen your net, you start to see things that don't line up with our previous expectations, because we are extrapolating from limited data. But within the reproductive season, let me quickly explain what we see as I move my face out of the way on the photo on the right, or the diagram on the right. So what we can see here, this is from early drafts of some work that we did. Imagine this clock. The midnight is FM, stands for full moon. And as you go clockwise, we break up into 6-hour increments. So that's one of the first things that one can do. You, rather than going from number of nights after the full moon and number of months of the year that corresponding is happening, you can start to be a little bit more precise at zero cost. How many hours after the full moon would corals be active? And so this is just an example where we can see that actually, if depending on the day and the year, this is from a few years of work, between 110 to 128 hours after the full moon, we had the greatest diversity of corals spawning. So each of these colors represents a coral that is actually redocumented via scuba surveys to be releasing gametes. And in many cases, most of these genera had never been formally documented to be reproductively active in the country. And this is just one snapshot. That a bunch of other work had been starting to show a similar thing. So you can start to be a little bit more precise with your timing. On the bottom is essentially comparing what are the general diversity of corals on your reef versus what is reproductively active on that reef. Ignore the ROV bars for a second. I'll explain that in a moment. But if you look at the baseline, the PIT baseline, that is a very rough assessment of here is the diversity of corals on one example reef. And if you look at the scuba spawn bar, that is the number of corals, including the number of colonies that spawned during a given period. Those bars are somewhat similar, but they do not match up. That means that there are many corals that are on the reef that we have no spawning information for. And there are many corals that are spawning that are better represented in their reproductive activity than how abundant they are in the reef. So that means that there is a lot of information that we do not have. And so it starts to motivate looking outside of the natural coral spawning windows to figure out what else might be happening. Just to address the ROV thing very quickly, on the eastern Gulf of Thailand, our coral spawning period lines up perfectly with Chironex, which is a genus of box jellyfish, for those that are unfamiliar, that has been the genus of the greatest number of fatalities in, I believe, the world from a box jelly perspective, certainly in Thailand. And it is an invisible creature that is hard to spot even with white light, which means that we start to have to make trade-offs between being in the water and seeing potential risks in the water where one can protect yourself. So sometimes we have to choose between effects on corals versus human health. So red light versus white light, these are the kinds of conversations that we need to figure out. We use an ROV to get in the water instead of us, because an ROV is famously immune to Chironex, we believe, yet hypothesis to be tested. So by reframing our understanding of how one can predict coral spawning, we can start to ask different kinds of questions. If we want to talk about diversity, if we want to start to investigate the corals that challenge the preconceived ideas that we've had largely from the Acroporidae, the Poeciloporidae, and the Poritidae, there are many corals on a reef that are not these. We would need to start to look beyond just the information that we have and look at what information we don't have. So I'm going to stroke my own ego here for a second to give you an example of this. Improving accessibility built on data that you can collect from in-situ observation allow for a great change in the capacity for a region to do this kind of work. What do I mean? So from some of our work that was not really spawning specific, that distilled many years of data from Koh Tao, an island in Thailand, we were able to identify coral spawning trends with a decent amount of precision. This is data that had been collected over a 9-year period, multiple genera, many species. And so working with different groups in Cambodia, I was able to draft literally just a 1-page Word document introducing the idea, introducing these are the things to look for at these specific nights at these specific times, and you should be able to see if it lines up, corals, if the corals are reproductively active. And lo and behold, Cambodia was able to document now their first, second, and as of earlier this month, third ever coral spawning period, coral spawning documentation in the country's history, and are now able to start taking conservation action thanks to this information. So this did not come from any histological work needing to be done in the country. This came from, here's a Word document, information that you can look at the trends that are being seen nearby and apply that. And theoretically, because your corals, are closely related to the corals in Thailand, they should all be doing a similar thing potentially, and that is how it panned out. So there's a lot of ways to scale and use data in ways that modernizes coral spawning documentation in parts of the world that capacity has not really been apparent as well. And obviously, I would say that the greatest success of this is the collaboration of Indo-Pacific Coral Spawning Database by Andrew Baird and more than 100 other folk that all pooled in their data to establish a baseline for coral spawning timing across the Indo-Pacific for a lot of different places, not everywhere, and for a lot of different corals, not every coral, but by far the richest vein to understand what do we know and what do we not know, right? And if you look at the word Pavona, spoiler, there is not a huge amount we understood and still understand. So daytime coral spawning. Over the years, snippets of observations have been made in the literature and occasionally on social media, but rarely convincingly, of corals doing something that looks a bit like sperm release at the very least, gamete release, at a time when the sun is still up, which is, I think most people would agree, daytime. And these snapshots allow us to understand that one of the biggest internal expectations within the community for coral spawning, that being that it happens at night, that's something that has largely been established for many, many years, starts to get challenged. Occasionally, it's sporadic colonies in sporadic locations, often unrelated. To be honest, it's often parietes and occasionally Pavona. And that really led to an inkling that there might be more going on. And I would argue that the best work of this came out last year through some folk that are in the audience right now that worked and investigated parietes roofs across a wide biogeographic range in the Pacific and identified predicted daytime, if I remember correctly, fairly early morning coral spawning of parietes roofs in the Southern Hemisphere. And so that work happened at the same time as the work that I'm going to talk about now, which I would say firmly establishes that some corals refuse to spawn at nighttime and they will only spawn during the daytime. And we should therefore expand our investigations into looking at the convenient time where we don't have to stay up before going to bed. So let's talk about the actual story from our work. This is not the first image of the daytime spawning, but this is an image that I most like to show people because it tells you a lot the more you look at it and the more you understand what you're looking at. Prior to our work, a couple of pieces of, from a prior team that I was involved in and through work from Ajahn Sakanan, who is a coral specialist here in Thailand, snippets of Pavona daytime spawning had been recorded. And then we incidentally found another mass spawning, I say mass, like 12 different colonies spawning in a 10-minute period right after lunch one day in December during the COVID years. And that really started to say, okay, there's something going on here. These are not just fluke events or confused corals. Keep in mind, corals can also be confused. And so we need to understand this. And so we pulled that information and we pulled our observations in together to hypothesize potentially a new predictive framework, which is, okay, it's not happening at night, it's happening at day, it's around midday, and it's happening at the completely wrong, I put it in inverted commas, time of the year. It's not in February, March, or April. This was in, this was in November. The first one was in November. So it's 6 months apart, half year apart. It's breaking all the rules. That's, if you're a marine biologist and you find something that breaks all the rules, nothing can be more exciting because now it's just a world of discovery. This image was taken in our second year of observations. We collected, we started to look for Pavona coral spawning in the daytime, and I'll go into more detail in a second. Between 2020 and 2020, well, we're still doing it, but up to 2024 and 2025 a bit as well. But the bleaching event had an impact. And in this image, it was the first time that we could understand a little bit more. We had been documenting corals, Pavona daytime spawning, very much more like the, what you see on the right half of this image. This is what typical sperm release looks like in corals. It is a cloud of diffuse, 50 shades of white material being released. And on the left, you see something that looks very, very similar. For those that are looking at this not on their phone or potentially on a good phone, you can zoom in, or for those looking on your screen, you might see that what's happening on the left is a little bit different. There are strings being released, very similar to sperm release, but it's almost looks pixelated. It's made up of tiny, tiny white dots. And we later found out that these are in fact eggs. And what you can't see is behind this colony of Briarium that is dividing the photo on the left and photo on the right, this is actually one colony. So this observation told us that what we had assumed to be a gonochoric incidental spawning that was happening from time to time, actually this colony is hermaphroditic. It is releasing eggs on one side exclusively and sperm on another side exclusively. And approximately 5% of the colonies that we observed over the period appear to be simultaneous hermaphrodites. All of the other colonies that we came across were either only releasing sperm or only releasing eggs, which tells us that it is predominantly gonochoric, but there is some hermaphroditic thing happening. But keep in mind, this is where the limitations of in situ observation come apart, because you cannot tell everything just from being in the water. You would need to do deeper, truly biological investigation to understand development and understand the proportions. We were extrapolating purely get in the water and see what happens and if your predictions are right, and taking that as far as we could. So very quickly, for those that don't know, this is the genus Pavona. It is a coral that is has a variety of, I think there's 19 species that are valid today. Could be wrong. And there's many of the most well-known ones are folios on the reef, and then there's some massive and encrusting ones. We're going to be focusing our story on Pavona explanulata and varians, because we have not found any information on Decusata, Frondifera, cactus, and the other Pavonas that we can find in Thailand. So, as I said, we started to see these incidental observations, and we started to map out the timing of the coral spawning and map out the locations of these coral spawnings and corresponding events, and tried to broaden our search. So we included sites in Cambodia, the eastern Thai part of the Gulf of Thailand, and elements of the historic data that we had from the western side. And let me just translate the diagram on your left. This is not a clock. This is not telling you the time of spawning as much. It tells you the number of hours after sunrise that spawning was documented. So midnight would be basically at sunrise. And 6 p.m. effectively would be sunset, or as close to that. And so we can see that throughout the years of observations, Pavona varians and explanulata consistently will spawn before sunset, well before sunset, halfway between sunrise and sunset, around the middle of the day or in early afternoon. And then we found a few other cases of the Dipsastria is a somewhat questionable observation. We did find it, but I don't think it was ID'd properly. But Cycloceres, we found a single case of multiple Cycloceres individuals, releasing gametes in January, again in the middle of the day. At a single location. So it starts to tell us that there is a lot that we don't understand. And this is why we've been investigating at the specific times of the year that we see the reproductive activity of Pavona. And we managed to pin that down, at least on the eastern Gulf of Thailand, for September and October predominantly, and occasionally November as well, or in some places, October and November predominantly, and occasionally September, very rarely December. Very different from the nighttime coral spawning. So let's contextualize that a little bit. The only way we were able to predict this and find this is because we wanted to break the conventional framing that a lot of coral spawning work had been done in Thailand prior, which is you're looking for the number of hours after sunset and days after the full moon. You're looking for coral spawning in a specific coral spawning season. You're establishing your information from gamete development, and therefore you're biasing your information to look within that window. But there are things that, as I said earlier, you can do to change that. You can look at the precise time of the day and precise time out of the night, and you can calculate the number of hours either after sunrise or after sunset. But the one thing I like in particular is the number of hours after the lunar peak, which goes back to that little diagram that I had a few slides ago. You can get greater precision of timing, because keep in mind, sunset and sunrise is not going to be the same as when the full moon happens. The full moon doesn't, the lunar and solar timing don't always need to synchronize. In fact, they rarely will. So the number of hours after the lunar peak can perhaps give you a bit more precision, and they ended up giving us more precision. And looking at classification and evolution. So simply put, are more closely corals related spawning at similar times? In many cases, yes. So for example, the nighttime coral spawning was dominated by Merolinidae taxa. So all of the genera in the greatest cause of complexity in a lot of coral ID folk. In the diagram on the right, what you can actually see on the upper part is the middle is the monsoon season. We couldn't get in the water. All of the light small gray dots, those are null data. This is one of the most important things I think one can do. Anytime you get in the water and you don't see coral spawning, daytime or nighttime, that tells you something. So we tried to compile all of that information to figure out when there was no coral spawning happening. And what we can see is the time of the year versus hours after sunrise. We reframe things to be hours after sunrise because we're talking about daytime coral spawning. On the upper part, we can see days in the new year. That tells you days from new year. So the time of the year, we can see that there's a big difference. On the right, we see the Pavona daytime spawning happening in the second half of the year, and on the first half of the year, where we see the nighttime coral spawning. And if you look at the bottom part of that plot, it's just looking at the lunar influence. And again, we see some clustering where in the nighttime coral spawning, they tend to be clustered around between 3 to 6 days after the full moon, whereas with the daytime coral spawning, it's anything from a couple of days before to a couple of days after the full moon. So the lunar signal that promotes the synchrony of coral spawning is behaving differently with these two species of Pavona. Keep in mind, these are just two. And keep in mind that because we keep having null data in place, had we seen other species of Pavona spawn, we would have documented it. We did not see them. And we did, in fact, go and look for Parietes roofs at the same timings that my colleagues went to find it. But we're in the Northern Hemisphere and we found diddly. Does that mean that they're not reproductively active? Of course not. But that means that they don't follow these two trends. So they must be spawning at some other time, whether time of the year or time of the day. And that's just one single species. There's tons of other corals that we have no information for. You will see the legend here does not include every single coral that one can find on an average Thai reef. Globally speaking, we wanted to contextualize what we were finding in Thailand and in Cambodia with other parts of the world. And we try to pull the reproductive inflow for all of the Pavona species across the world, obviously focusing on Explainulata and variants in particular. And it does appear that there is some sort of a geographic influence in terms of the coral spawning timing, but nowhere else is seen synchronized daytime coral spawning in Pavonas like we are, even in theoretically the same species. Now, of course, there are questions of whether the Pavona varians in one part of the world is the same as what we have here, Explainulata, so on and so forth. Pavona is still TBD on a big taxonomic revolution that others are going through, and I'm excited to see that. But till then, we're working with what we understand right now. And globally, we seem to have the same species as a few other locations, but only ours seem to be spawning in the daytime. But it is synchronized, and it goes across more than 400 kilometers. We think probably double that if you include the Western Gulf. So it is synchronized across a very large geographic range, but it seems to be a phenomenon that's focused in the Gulf of Thailand. For now, I would love to see nearby regions investigate this, and I'd love to see what happens in the Southern Hemisphere as well, because if you can't already tell, the whole point of this talk is there's more we don't know than what we know, and that's okay. So next steps, I hope I'm doing okay with time. I will try to be fairly quick with this. This is some of our most recent work. So we have been trying to track the dispersal of our coral spawning events. This is currently unpublished, and this is fresh, hot off the, well, not even the press, hot off my data of the tracking of where dispersal might happen or seems to be happening across the Gulf of Thailand. And it seems like it doesn't always agree with the general nationwide level models, and you need higher spatial resolution to understand how some parts of, in this case, a country or a Gulf can be connected from one another. So we can see, for example, that Vietnam and Cambodia in some times of the year will get coral larvae coming from Thailand. And in other times of the year, for example, with Pavona, we might be seeing very different patterns. That means that your reefs are interconnected and they don't care about passwords. There is a lot of more connectivity across the region in theory, and there's a lot more work we need to do about that. But tracking coral spawning will also point at one very, I would say, crucial point. When you're looking at juvenile corals on your reef, there's a lot of assumption that when we restore a specific reef, we are protecting that reef long term. What you're doing is you're promoting corals for whatever reef is downstream. And the future of your reef largely depends on what's happening upstream. And from a conservation standpoint, I'm biased, I would call that essential to think about. So linking coral spawning to conservation, this is something, if you will indulge me, I would like to talk about for a second. Understanding upstream and downstream reefs and dispersal patterns during coral spawning can tell us a lot about which reefs can have juvenile corals into the future. And that also influences how one can work with policy and zonation. More interconnected conservation management would benefit not just local reefs, but ecosystems that are connected hydrodynamically, genetically, and in other ways. From a restoration standpoint, you need successful coral spawning to bring in new genetic material into a reef. If coral spawning does not happen, and more importantly, if fertilization doesn't happen successfully, and those fertile larvae and those fertile juvenile corals don't reach your reef is only sustaining itself based on asexual reproduction. And well, there are issues with that because then your reef is on a bit of a ticking time bomb in some sense because we have large-scale mortality events. I imagine everybody in this particular chat is aware of. This is my favorite visual reference of talking about this. Disturbance events, especially when we talk about things like succession, the greater the diversity you have, whether it's the diversity of species, whether it's the diversity of genomes within a population, the greater the diversity you have, the greater the resilience you have. Because if there is a threat, of course, can affect everything. Coral bleaching does not affect just one coral, but certain threats can often be affecting certain communities within a population more than others. Whereas if everything becomes monospecific, you can ask the terrestrial world of conservation and science, monocultures tend to be not so great. And again, we can talk more about that later if you'd like. I want to use us as an example for a very quick second to illustrate this point, not to scratch our own back, but to tell you all the things we did wrong. Well, I say did wrong. The ways that we, all of our failures and what we learned from our failures. We were encouraged to do active restoration as part of our work for a few years. We did a direct attachment with epoxy. We also put down substrates to study recruitment. We use everything from fishing lines and epoxy and all of the techniques that I'm sure everybody in this call are familiar with. And we had a bunch of success for a number of years. Here you can see everybody's favorite Acropora. We did that because our acros were in desperate need of help, but we also focused on Poslepora. Many parts of the world, Poslepora is a ridiculous coral to want to restore because it is the weed that grows everywhere. But for our archipelago where we were at, most possels had kicked the bucket before acro, and for reasons we don't fully understand. So things were going great for a number of years. This was just one colony over 1 year. Then in 2024, I'm sure everybody knows, things kind of got warm and we had just a large majority of our corals, hundreds of corals that were part of the active restoration work that we were doing, die. Not just Acropora, multiple genera, but acro in particular. Our acros were natural fragments across multiple species and multiple colonies over several islands range. So we tried to maximize diversity, both taxonomically and in terms of genetics. And so we did see some survive the bleaching event. We had more survival than the acros on the natural reef, which were, 70+% in many places. So very quickly, a huge amount of our corals did not survive. And I wish more people would speak about the techniques that they have used that did not encourage long-term survival of their corals. Natural reef, again, this is just more for illustrating the point. We had pockets of tissue still left on many corals that had been healthy just a few months prior, and the rest of the colony, meters or centimeters, however that might be, not surviving. So we had large amounts of mortality on the natural reef in 2024, just like in 2020, just like in 2016. Again, not beating that home. I do want to illustrate one point. On artificial structures, and in some cases on the natural reef, juvenile corals, and so through the process of coral spawning, reproduction, and coral recruitment, juvenile corals were bleaching less, surviving more, dying less. And we found this across the board. So on the right, what you see is concrete structures at one particular location. These photos were taken on the same day. And the proportion of bleaching is far less, and the proportion, I can tell you later on, basically the proportion of survival was night and day. We had less than 10% mortality across all genera on the artificial structures. This is not anybody transplanting anything. This is the Thai government put down a bunch of structures because they could, because of well-informed or not decision-making, and it worked based on whatever goals they may have had. And of course, we had some of our active restoration corals survive as well, but juveniles, natural recruits, that some of them had been very, very young at the start of the coral bleaching, some of them were maybe a year old at this point, mostly seem to survive. These are on some of our structures, not the government structures. And yeah, we found, as I said, I'm just driving that same point home, far, far less mortality and far greater survival. Basically, we got about 5-point-something% mortality of acropora on the recruitment side, and we had 70+% on the natural reef at the same site. So here are some acros that I'm surprised did not die. Yeah. So that, I hope, emphasizes not only coral spawning and the discovery of new coral spawning patterns, but the value of coral spawning and sexual reproduction in many places. While this applies to Thailand, there are many parts of the world where coral recruitment failure is the story. They are having large scale loss of coral recruitment and lack of survival and a lack of diversity of coral recruitment across their sites. So this is something that is working for us here, may not work everywhere, but it is something to keep in mind. Coral spawning events themselves are synchronized in one form or another, but they don't all happen at night. They don't all happen at the same time of the year. And it is important that we expand our hunt and keep track of every time we don't see something. I promise that every 0 you write down has more value than you realize. And that coral spawning, understanding coral spawning, the ecology of coral spawning, and the implications of coral spawning in coral conservation is a much closer link in many places than perhaps people realize. And again, I'm preaching to the choir here, I think, but sharing our story a little bit. Thank you for putting up for me rambling for as long as I did. I hope I didn't go too overboard. Yeah.